Hello, I'm Stephen Turner, and I'm a principal bioinformatics engineer at Form Bio. Today I'm going to show you how to use RStudio and Form Bench. Form Bench is a way to launch an application running on the Form platform using data in your project, and RStudio is one of the applications that you can run using Form Bench. And the reason you might want to do this is, let's imagine that you've done some analysis on the platform using one of our pre-existing workflows, or maybe a workflow that you brought yourself, and maybe you want to take that a little bit further and really dig into some of the results or data that you have and maybe produce some visualizations or do some other downstream analysis that might be custom or unique in some way where there isn't really a workflow that accomplishes what you're trying to do. And so today I'm going to show you a statistical analysis of transcription factor binding site disrupting SNPs in a set of genes. And today I'm going to use a package called tfboot. This is an R package that we published in collaboration with Colossal Biosciences. The preprint is on bioRxiv, and from bioRxiv you can click into the paper, and when the paper loads you can click into the PDF, or you can click directly from the abstract page, but you can click into the source code and take a look at the actual code. This is open source, it's MIT license. You can get some installation instructions. It's installed the same way any other R packages from GitHub. And there's a package down documentation page with a couple of vignettes. And what I'm going to show you here is largely taken from the vignette that is on the public package down page that's also published with the paper itself. So back to Form Bench. Before I actually dive into Form Bench, I want to take a minute to show you about the Files pane for a minute. So if you click into the Files pane, these are files that you have uploaded into your project already. And so if you look, there's a folder called Form Bench Demo RStudio that has a couple of files in it. And so you'll see how these tie into the RStudio server instance in just a minute. So if I go to Form Bench and I go to Launch, RStudio, give this a name, tfboot. I can pick what kind of instance I want to run this on. In this case, I'm just going to use the default of a 32 CPU, 128 gigabytes of RAM. I don't need a lot of space or time, so I'm just going to launch that. Now this takes a couple of minutes to launch. I'm going to go ahead and hop into one that I've already got running, that I launched a couple of minutes ago. This takes about 1 or 2 minutes to launch. And you'll see when you pull this up, it looks just like RStudio running on any other server that you might have RStudio running on. You'll see that my home directory just has this files folder. And when I click into that, you'll notice that you see the same things that I saw back in my file browser on the Form Bio platform page, specifically this Form Bench demo RStudio folder. And so in that, I'm going to click into that and launch the project, which is going to open RStudio server running in that folder. And you'll see when the project launches running in that directory, you'll see some of the files that I already have in there. And so I'm going to pull up an R script that is kind of a pared down version of what's in that vignette and show you kind of walking through an analysis. So the first thing that I'm going to do when I open up this instance is I'm going to install a few packages. So I'm going to install DevTools because I need that to install a package from GitHub. I'm going to install BioC Manager because I need that to install some packages from Bioconductor. I'm also going to install Tidyverse because everybody uses that everywhere, and Markdown because I'm going to compile an R Markdown report in just a minute. So I'm going to go ahead and install that, and you'll see as this is installing that it's actually pulling from the POSIT package manager. So you'll see instead of pulling from CRAN and installing from source, which could take a long time for something like the Tidyverse, which installs many packages, this is really quick because it's pulling binaries from the POSIT package manager. So I'm going to skip over installing some of the Bioconductor packages because they're already on this instance. I'll just demonstrate really quickly installing from GitHub. This package is not on CRAN yet, but I can install this from GitHub using DevTools. And so you'll see when it does that, it takes a minute. It pulls the source code, and then it actually does compile and build from source because there is no pre-existing binary available. So once all those packages install, I only need to do that one time. I'm going to load all of those packages at once because I'm going to use almost all of these packages throughout the demonstration here. And once I've loaded the tfboot library and others here, I could get some help on some of the functions in the tfboot package, just like I would get help on any other function in any other package. In this case, I'm going to get help on the motif maker bootstrap function that's in tfboot, and so I can get help on that. This is coming from the tfboot documentation. I can actually click into here and it'll run the actual examples from the package and sort of like a little subshell kind of thing. So let me go back. So to actually kind of walk through this analysis, what I'm going to demonstrate really quickly is first I'm going to read in a VCF file. I'm going to extract the SNPs from that VCF file. I can take a look at that. It comes in as the GRanges object. And the next thing that I'm going to do is get SNPs that are upstream of, or get regions that are upstream of these SNPs of interest using a combination of those SNPs and a TXDB from the same organism. So this is some data from chicken. So I'm just going to take a look at those, more genomic ranges. The next thing that I'm going to do is get a selection of 5 genes of interest that I want to test for statistical significance of transcription factor binding site disrupting SNPs in those genes. And so if I take a look at those, these are just the gene IDs. I can look at the annotation database to see what they actually are. Here are those genes. And so the first thing I'm going to do is I'm going to pull out SNPs around the promoter regions of those SNPs. So if I take a look at those, these are SNPs and the promoters of those. I can get some stats on those. These SNPs on average have, about 12.5 SNPs in the upstream promoter regions of those genes. And so the next thing that this thing does is, if pre-existing data does not exist, what it's going to do is use the JASPAR 2022 transcription factor binding site position weight matrix data motifs, and it's going to run motif breaker on that small set of genes and on every gene that I have, in this case, which I think is on chromosome 33, I believe. And then it's going to convert those all into tibbles, and then it's going to save that out to this pre-existing data file. That I've already done here, and that already exists in my files for this project, and so this should be really quick. It's going to check to see if that exists, and if it does exist, it's going to load that into working memory. This step takes about 5 minutes, so I'm going to skip that and just load the preloaded data, or pre-calculated data. I could take a look at the actual data that got loaded in here, so it tells me here's the gene ID, the SNP, and the various metrics that come out of motif breaker. So I'm going to run one of the tfboot functions on here to get a summary of the motif breaker results. The next thing that I'm going to do is create a bootstrap resampling of the results on all of my data. And I'm only going to use 250 bootstrap resamples here just to kind of keep things kind of quick, but normally you might want to do this 1,000 or 10,000 times. And I can take a look at that data. And everything that's being shown here is explained in great detail in the vignette. Finally, I'll get some statistics on that, and what comes out of this is comparing the motif breaker results on my set of genes of interest against a bootstrap resampling of genes across the entire genome of the same set size, and it's giving me p-values showing me the statistical significance of various different scores that motif breaker outputs. And finally, I could plot those and look at those visually. And none of these are particularly interesting. As we expect, this is a random subset of genes. And finally, I could get my session info. So this is just an R script. I've got all of this code in a markdown document, so you can get one of these by going to File, New File. And I copied over all of the code and put in some commentary here and there. So this is the exact same data, exact same code. I can pull up that, I can knit this R Markdown document, and a couple of seconds later, we should get a preview of an HTML file that we could download out of Form Bench and send to a collaborator or send to another interested party. So here's the HTML file that was produced by the R Markdown document that I just showed you, and this is again demonstrating all of the analysis that we just showed. The last thing that might be of interest here, you can run R code, R Markdown documents, Quarto documents, Python, etc. You can also click into a terminal. So this is a way to actually shell into this instance that's running on the platform. So if I just take a look at the data that I have here, it's all of the data that exists in this folder. I can back out and go into Files and see what else is here. And I could do other stuff on the command line that I wanted to. If I wanted to launch a text editor, I could do that. I could apt-get install something or do anything else that I would want to do with the terminal.